Well, thank you, everyone. Welcome to our August California Botanical Speakers Series at the California Botanic Society. My name is Mitchell Coleman. I'm the membership chair on council here. You may get random emails from me, but I'm really excited to introduce Dr. Helen Holmlund. This is the first person I've invited personally for this speaker series, and thank you, Helen, for accepting. Really excited to hear your talk. I'm just gonna be safe and read her bio. So just quick introduction. Dr. Helen Holmlund is an assistant professor of biology at Pepperdine University. She earned her BS in biology at Oklahoma Christian University and her PhD in ecology and evolutionary biology at the University of California, Santa Cruz. During her graduate work, she also spent a year training at the Australian National University. Her research in the field of plant physiological ecology explores how plants can survive drought, fire, and salinity. Oh yes, salinity, yay. In particular, that's not a good thing in salinity, but it's an exciting research field. In particular, oh, my screen darked. In particular, Dr. Holmlund is fascinated with ferns, which typically require abundant water, and how some ferns have adapted to thrive in dry ecosystems, such as the Santa Monica Mountains in Southern California. Her research on these extreme ferns has also taken her to the Channel Islands, the mangrove swamps of Australia, and the tropical rainforests in Costa Rica. So it's a really cool research field. Dr. Holmlund, we are extremely excited to have you. I'll go ahead and turn it over to you.
Wonderful. Mitchell, thank you so much for the introduction, and thank you all so much for the invitation to speak. I'm so excited to share my research with you. Can you all see my screen all right?
Yes.
Awesome. Wonderful. Thanks so much. Well, like Mitchell said, my name is Helen Holmlund, and I teach plant biology and ecology at Pepperdine University. My research focuses on plants in the chaparral ecosystem that surrounds us here in Southern California, and it is one of the most biodiverse, drought-adapted ecosystems on the planet. And yet, we still have so much to learn about the plants, and especially the ferns. Here you can see a fern growing in the chaparral, and it looks dead, but actually it is still alive. Let me just start by giving you a little background about my journey as a scientist. I am a plant physiological ecologist, which means that I like to study plant physiology or plant function within this broader ecological context. I'm also really interested in questions about how structure or anatomy relates to function or physiology. I first came to Pepperdine as an undergraduate research student, but I have recently started working here as a professor. Well, the chaparral, which covers the mountains around Pepperdine, is a dry ecosystem that is dominated by drought-hardy shrubs. So Pepperdine is a really great place to study plant adaptation to drought. This ecosystem is local to our area, but it scales to the whole globe because there are 4 other ecosystems with a similar climate and group of plants, all called Mediterranean-type ecosystems. Besides California, these ecosystems include the Mediterranean region, Chile, South Africa, and part of Australia. These ecosystems are especially interesting because they are hotspots of biodiversity where you have a lot of different plants growing in a small area. These ecosystems are also potentially threatened by climate change as things get hotter and drier, as we have already seen on Pepperdine's campus with plant dieback due to drought, fungus, and frequent fire. So, lots of folks have studied the shrubs that grow around Pepperdine, but the ferns are another group of plants that thrive here. These ferns are also drought adapted to survive in this dry ecosystem, but almost nothing was known about the chaparral ferns. So that was the question that I posed for my first research project at Pepperdine. How do the ferns survive here? And you know, this was my first research question as an undergraduate research student. And essentially, you know, throughout grad school and starting as a professor, I really just never finished that project. It just keeps going and it's so much fun. Though, of course, to be fair, first of all, you have to ask the question, why study the ferns? Well, ferns are major players in a variety of ecosystems, including the redwood forest and the jungles in the tropics. Most ferns in the world grow in places like these where there's lots of water. Some people then assume that ferns can only grow with a lot of water, but that's not true. Ferns also thrive in dry places like the desert. They might be small and growing in places that you don't expect, but they're there and they're part of these ecosystems. Studying drought-adapted ferns can tell us new things about how plants handle drought. So throughout my career, I just became really inspired by this question on how ferns growing all over the world have adapted to survive with very little water. In grad school, I explored other drought-adapted ferns. For example, I was given a grant to spend a year in Australia studying the mangrove ferns. Mangrove ferns have a lot of water around, but it's all salty, so they're adapted to thrive with limited fresh water available. Here I am measuring photosynthesis on mangrove ferns while working in a boat, so I don't get eaten by the crocodiles. In Costa Rica, I assisted with my advisor's project on ferns that grow on top of trees called epiphytes. Epiphytes are also likely drought adapted because they have limited access to water growing all the way up there on those tree branches. Sampling epiphytes is a bit tricky though. Here you can see me in this picture at the top of the tree. However, as exciting as those ferns are, I think that if you could study ferns anywhere, the most exciting place would be right here in the chaparral in the Santa Monica Mountains. Every year, the ferns here survive a 6-month summer drought, and some years it's even longer. Despite this challenge, lots of fern species thrive here. So my question was, how do ferns survive seasonal drought? To answer this question, I measured how the chaparral fern species use water differently. To give you a taste of what we found, let me show you a picture of 4 fern species growing side by side in the same habitat. Here the species are numbered 1, 2, 3, and 4. These species are growing side by side, but what we found is that they're using water in really different ways to survive. Given that they're in the same microsite, they are in one sense competing for the same resources, but what we found is that they seem to be partitioning resources and filling separate niches, and I'll show you kind of how that happens. First of all, these ferns have different lifestyles. This first one here is an evergreen fern, Dryopteris arguta, the coastal wood fern. And evergreen is just what it sounds like here. It maintains green leaves all year round, but that's incredibly remarkable to maintain a green leaf all year round when you have a 6-month summer drought. So it's pretty tough in that regard. The second fern, Adiantum jordani, a maidenhair fern, is deciduous. That means in the summer drought, the leaves die and fall off and blow away in the wind, and next year it grows new leaves. In a sense, then it sort of escapes the summer drought because it doesn't have green functioning leaves that are suffering during the dry summer drought. All right, my favorite last are these 2 resurrection fern species. You have Pelea andrometifolia, the coffee fern, and then Pentagramma triangularis, the goldback fern. They're called resurrection ferns because during the summer, those leaves dry up and look dead, and they're like dead looking and crunchy. And when it rains, the same dead looking leaves uncurl and turn green and come back to life. So they also sort of escape the summer drought by not having living tissue or metabolically active tissue during the summer, but they do it in a very different way. And to kind of illustrate the life cycle of these 4 species, you can see them here in April, and then here they are in June. As you can see in June, just 2 months later, that evergreen fern is still green. The deciduous fern, those leaves have dried up and they're dead and they're going to blow away and fall off. And the resurrection ferns are curled up so tightly and so compactly that you can barely even see them. So the thing is, they're growing side by side, but I wanted to explore further how to what extent are they really competing with each other for water? And it turns out that they have different types of drought tolerance. And I measured a variety of aspects of these ferns. First of all, I looked at how dehydration tolerant the leaf tissue is. The y-axis here is showing the dehydration tolerance or the water potential in megapascals, and it goes from 0 megapascals, which is perfectly hydrated, perfectly happy, to minus 10 megapascals, which is incredibly dehydrated. To give you perspective, most of your crop plants or house plants are probably suffering by minus 2 megapascals or so. And this is showing you on the x-axis the same 4 species. And this is the most extreme water potential that each species can handle before its leaf tissue dies or goes dormant. So right off the bat, we see that there are differences among the 4 species. And this evergreen fern on the left, Dryopteris arguta, is an incredible champion. Its leaves are so tough, so dehydration tolerant. To my knowledge, having leaves that can stay alive or in functional at -8 megapascals makes this fern the most dehydration tolerant fern ever reported in the literature. Now, the second fern is deciduous with less dehydration tolerant leaves. At -3 megapascals, the leaves die and fall off. Now, by contrast, the resurrection ferns have even less dehydration tolerant leaves. They don't even make it really to -3 megapascals. However, that's okay because instead of dying, their leaves go dormant at -3 megapascals and get resurrected later when it rains. So the important thing to remember is that when the leaf completely dries out, the 2 of these species die and these 2 do not. Instead, they go dormant and then resurrect later on. When we were talking about the most negative fern water potentials that can be sustained while staying metabolically active, it would be Dryopteris. However, if we were talking about the most negative water potentials that a fern can survive while dormant, it would be the resurrection ferns. But the big takeaway here is you have 4 ferns, but 3 totally different strategies with regard to water use. But that's just one way that these ferns are different. Let me show you a couple other ways. Another thing that's interesting is these ferns, even though they're growing side by side and competing for water, they're taking water from different places in the soil. This is the root depth of these 4 ferns. And as you can see, the evergreen Dryopteris arguta has the deepest roots, which means it will be getting the most water during the summer drought. Now that makes sense that it has the deepest roots because it needs to sustain those leaves all summer long to be evergreen. The deciduous has shallower roots, which probably lets it dry out sooner. What was initially a surprise to us is that the resurrection ferns have really shallow roots, but then we thought about it some more, and that kind of makes sense. For these 2 resurrection ferns, there's not really a good reason to stay hydrated for a long time because being dried out and dormant, that's safe for them. So probably having shallow roots facilitates drying out quickly and getting to that safe, dried up, dormant state. Finally, we found differences in the photosynthetic speeds of these 4 ferns. You can sort of think of Dryopteris, the evergreen fern, is slow and steady, like kind of the tortoise and the hare, versus the other ferns have faster photosynthetic rates. And honestly, that kind of makes sense as well. The evergreen has all year, since it's evergreen, to conduct photosynthesis and produce sugars for itself, while the deciduous and resurrection ferns, they go dormant during the summer. So they have a shorter period of time to conduct photosynthesis, and therefore it makes sense they might have adapted to have faster photosynthetic rates. All in all, what's cool that these data show us is that niche segregation seems to be driven by a partitioning of water resources, which to us seems to make sense because water, to our knowledge, is essentially the most limited resource in the chaparral for these ferns. Well, so these ferns are incredibly exciting. For the rest of today, I'll focus on my favorite group of these ferns, which to me is the most remarkable strategy, which is the resurrection ferns. These resurrection ferns, they die out all summer long. Here you see the fern with its curled up, almost no free water leaves. And when it rains, the same dead looking leaves uncurl and turn green and come back to life, as you can see here. So then kind of the next set of questions I have is how does resurrection come back to life? Well, let's talk a little bit about what exactly are resurrection plants. Resurrection plants are desiccation tolerant, which means they can survive equilibrium with dry and then resurrect when rehydrate. Excuse me. They can survive 10% water content or minus 100 megapascals, which is incredibly dry. And most plants are desiccation tolerant at the seed, pollen, or spore stage, but the rest of the plant typically is not desiccation tolerant. So plants are non-vascular. The resurrection ferns that I study are true vascular plants. They have complex tissue types that vary in function, composition, and also they have to restore hydraulic through the vascular. So I asked the question, how does a dedicated vascular, such as a fern, resurrect? How is hydraulic flow restored? So I took this broad overarching question and I answered. First of all, use targeted irrigation to understand what water resources are most important for resurrection. Essentially, how is water moving through a resurrection fern when it comes back to life? Is it mostly coming from the roots or the leaves? Next, I analyzed the sugars, different tissues while they were resurrecting to get an idea of how the plants were using and storing energy. Finally, I used a micro CT scanner to X-ray the ferns while they were drying out and resurrecting to see how the water was moving through different tissues during that process. But first of all, let's take a look at my targeted irrigation experiments. We have two really abundant desiccation tolerant fern species at our study site in the chaparral. First of all, you have Pelea andrew. It rained 0.2 inches after 9 months of drought. Doesn't very much rain. So the ferns only resurrected partially. As you can see in coffee fern, the bottom half of the frond, the leaves resurrect, turn green and uncurled, while the top half is still dried up and desiccated leaves. So it only resurrected halfway. In the goldback fern, you can see the short leaves are uncurled green down here, or shorter leaves resurrected. And these short leaves were closer to the root water source. We thought that the root water instead of leaf was the primary water tissues. And this idea is based on how I normally think about water flow through a plant. A lot of water to the leaves where it evaporates out the stomata. Thus, root water is normally the major supplier of water to the plant. However, there is at least one other option, which is that leaf water might resurrect the fern. When it rains, all the leaves get wet, so perhaps that's a faster way to resurrect. Thus, we need to consider the possibility leaf water might be the primary mechanism of resurrection in these ferns. So this led me to propose. First of all, based on our resurrection of our observation of partial resurrection after rain, only the leaves closest to the roots resurrecting, we hypothesized that root alone can resurrect the plant. To test this hypothesis, I'm out in the field during the summer when all the ferns were dried up and there'd only measure leaf, abbreviated FEFM, also known as maximum quantum yield, which is a measure of class function. To look at root recovery, I measured root pressure, which is a phenomenon in some plants where the roots actually push water into the rest of the plant. And we observed that in these resurrection ferns. I measured root pressure with bubble manometers in the field. So here is a resurrected pentagramma plant that I photographed. By the way, this is a real plant, and you can see that it has these short roots, which we think are good for fast desiccation and fast resurrection. But when I watered the roots, found that there was root pressure. So that indicates that the roots were active. So the roots did recover. And I found that the leaves recovered as well. Now it took a while. It took a while for the root water to get all the way to the leaves. It didn't really resurrect until about day 8 or 9. But nonetheless, that led me to conclude that root water is capable of resurrecting whole plant in the field, from the roots to the leaves. my next hypothesis was that leaf water alone can resurrect the whole plant. In that experiment, I sprayed leaves of plants in the field containing stalking roots dry. That was a lot of running around in the bushes and spraying the leaves. And this time I found that the leaves resurrected faster. And honestly, that makes sense because I was applying water directly to the leaves. So it makes sense that the leaves would resurrect faster. However, the roots never resurrected. There was never root pressure in the time of that experiment. So this led me to conclude that leaf water only resurrects. Next, I hypothesized that leaf water even leaf water alone isn't enough to resurrect a plant. Maybe leaf water and root water together resurrect the plant faster than root alone. And this makes sense because if you're a photosynthetic organism, then time is carbon. And it makes sense that these ferns might have adapted to use that leaf water resource. In this experiment, I covered one of the leaves of some plants during a rain event to keep some of the leaves dry, and then compared wet leaf and dry leaf treatments. So here you can see from my diagram, before a rain event, I covered the leaves of half the plants, and those plants only need water to this. Applied pressure was needed for leaf recovery, all with standardized stipe length, but the leaves only recovered when pressure was applied, and it didn't recover when capillary action alone was working. Now, this is a case where there is a difference between the two resurrection fern species. So let's take a look at Pelea, which has a more complex frond morphology. Again, this is the coffee fern. We found that applied pressure was still needed to resurrect the top leaves. So when you just stuck the whole frond in a tube of water with capillary action alone, you didn't really get much recovery, if any. But when you pushed water into the cut into the stem, that water then did make it all the way to the top leaves to resurrect them just a bit. However, the bottom leaves, the ones closest to the cut into the stem, they resurrected either way. So capillary action was enough to get to the bottom leaves, but just not enough to get all the way to the top. So what that showed us is that applied pressure was needed for recovery of the top leaves in this species. Well, that was kind of a lot, but just tying all of these experiments together, we made several conclusions based on our data. First of all, it appears that root water does contribute more to whole plant recovery than leaf water. This conclusion is based on the data showing that root water resurrected the whole plant, while leaf water did not. However, we did see that leaf water expedited the recovery of the leaves, which makes sense that these plants would have adapted to take advantage of that leaf water resource. And finally, based on our applied pressure experiments, it appeared that root pressure might be necessary for leaf resurrection in pentagramma and distal leaf resurrection in Pelea. This would be consistent with our findings of root pressure following irrigation or rain in the field. Well, next I wanted to know how energy is being used and produced during the desiccation and resurrection processes, because that might also contribute to the mechanism. Because, for example, one of the known mechanisms of desiccation tolerance is that sugars or proteins can surround the cellular components in a resurrection plant and protect them and stabilize them when the water is gone. So sugars might be a really important part of the story. So I looked at that next. But first of all, let's talk about what exactly I mean by non-structural carbohydrates. Essentially, they are just sugars that aren't mechanically supporting the plant. These include starch, which is great for long-term energy storage, and sucrose, glucose, and fructose, which are more immediate energy supply. To measure this in the resurrection ferns, I collaborated with Doctors Anna Sala and Gerard Sapez at the University of Montana. We used an enzymatic approach to quantify the amount of each type of sugar in each plant tissue while the plants were resurrecting. This is an especially interesting question in the resurrection ferns because some of those sugars, and especially sucrose, are known desiccation protectants to stabilize the cellular components of the plant while the water is gone in what's called the glassy state. So sucrose might be an especially important player. For this experiment, we focused on Pelea, the coffee fern, because it's bigger and we needed more plant material for the sugars analysis. We looked at sugars in several parts of the plant. Of course, we looked at it in the leaf tissue. We also looked at it in the rhizome tissue, which is in a horizontal underground stem where often long-term starch is stored. And we also looked in the petiole, but specifically, we looked in the vascular bundle of the petiole called the stele. That's where the vascular tissue is moving water up and flowing and sugars typically down. So looking at these three tissues, I looked at starch, sucrose, and glucose and fructose concentrations while they were resurrecting. Let's start with the leaves. On the y-axis, you have the percent dry mass that the given sugar is of the, and then you have the days following the rain. In the leaf tissue, what we found is that was day 0 being totally dried up. So that's a desiccated state. The different colors are different types of sugars, with the starch being dark gray, sucrose medium gray, and glucose and fructose being light gray. And note too, our starch analysis couldn't distinguish between small and large starch molecules. So most sugars of 3 or more units would have been grouped with starch. Much to our surprise, we found no significant changes over time in the leaves, and the glucose and fructose concentrations overall were very low. In the stele, that middle part of the petiole, we started to see some patterns. First of all, sucrose was highest in the desiccated state. That makes sense because sucrose has been known to act as a desiccation protectant for resurrection plants. So maybe high levels of sucrose were protecting the totally dry tissue. Interestingly, on day 2, there was a small peak of increased glucose and fructose. We don't know exactly what this could mean, but it's kind of exciting because it might indicate some sort of metabolic activity associated with the resurrection process. We really need more research to figure out what's going on there. Finally, looking at the rhizome, the underground stem that is typically used for sugar storage, we found some similar patterns. So like the stele, we see an increased sucrose in the desiccated state, which again could indicate desiccation protection in the glassy state. Again, like the stele, we see a small peak in glucose and fructose on day 2, which could indicate some sort of metabolic activity. And finally, we see a steady increase in the starch storage, consistent with the normal role of starch storage in a rhizome and perhaps returning to that normal function after rehydrating. So perhaps when it desiccates, that starch is converted to sucrose as a desiccation protectant, and then as it's resurrecting, converted back to starch. Well, kind of what we've learned from this particular set of experiments, it seems that desiccated steles and rhizomes are rich in sucrose, perhaps again consistent with that being a desiccation protectant. Those sucrose levels do drop early in recovery and appear to be converted to other types of sugars. And in the rhizome, starch stores rise in the week following resurrection. But of course, obviously, we still have some more questions because there's a lot going on inside the stem of a resurrection plant. I'm curious, inside the stem, what are the dynamics of recovery? Which tissues resurrect first, and do they help other tissues resurrect? And of course, what anatomical traits contribute to the resurrection through the stem? In other words, how does structure relate to function? And of course, to answer this question, I turned to advanced imaging to use the micro CT. For this project, I had the great honor of working with Doctors Anna Jacobson and Brandon Pratt at CSU Bakersfield. Micro CT is a really powerful tool to visualize water movement through plants because you can see what's inside the plant without cutting it open. Here you can see a picture of a dried out goldback fern. So, you have the dried up little leaf on top of a narrow petiole. And next to it is a 3-dimensional reconstruction of that petiole, of that stipe. And the signal is based on density. So the air shows up as black, and the water or solid tissue shows up as white or gray. From this three-dimensional reconstruction of the stipe, we can extract a two-dimensional cross-section to more clearly see how tissue sizes change with dehydration. Just to orient you, here is a light micrograph of a cross-section of the stipe, and the different tissues are readily apparent. The cortex takes up most of the stipe, providing support for the vascular bundle in the middle, called the stele. Normally, the cortex functions in water storage and mechanical support for the leaf. The stele includes the xylem and phloem and some parenchyma tissue. Here's a close-up of that stele, where you can see the xylem tubes, which transport water. You can see them over here, by the way, next to the smaller phloem and parenchyma cells, which transport or store sugars. My hypothesis, based on what we know about water movement through plants, was that the xylem tissue would be the first to desiccate and then the first to rehydrate. So to test this hypothesis, we imaged these ferns while they were drying out and resurrecting. So here is one of those images, because we would extract two-dimensional images from the three-dimensional reconstructions for an easier comparison. Here, for example, is a stipe on a hydrated plant that has never been desiccated. And just as a reminder, here is a light micrograph of the stipe. We can see some of the same tissues in both images. The stele includes water-filled xylem, hydrated phloem, and hydrated parenchyma. Since the water and the plant tissue have a similar density, all the tissues in the hydrated stele look gray. However, notice that the cortex is air-filled with those dark black spaces. We were surprised by this because usually this tissue is hydrated in fern stipes, and also these plants had never been dried out. So presumably the cortex must have dried out at some point in development, or else it dried out at a relatively high water potential. As the plants dried out, this is as we're drying out, air bubbles, air embolism became visible in the stipe xylem. Now, for most plants, gas embolisms in the xylem and the vascular conduits are a bad thing because air blocks the flow of water from the roots to the leaves. However, for these resurrection ferns, it is simply the first step to a desiccated, dormant state. Later in desiccation, more of the xylem conduits became gas-filled, but also the phloem and parenchyma began to shrink. You can see the shrinking phloem and parenchyma around. And as the phloem and parenchyma shrink, an air gap forms between the phloem and the cortex. Once the ferns reach that fully desiccated state, the phloem and parenchyma are even more compressed and that airspace is larger. And you can see the xylem conduits, they all kind of had to shrink and change shape as well. Now, let's look at the resurrection process. And you'll see the resurrection process is pretty much the reverse of the desiccation process. As the fern starts to resurrect, the first thing that happens is the phloem tissue appears to start to expand, which was a surprise to me because I hypothesized that the xylem would resurrect first. Later on in resurrection, you can see the xylem conduits refilling and those air bubbles disappearing. Now, when they were fully resurrected, most of the resurrected stems looked just like panel A. But we wanted to know whether the xylem was just filled with water or if it was actually moving the water. So we fed iohexol into the base of the stem, and iohexol is an X-ray dense molecule that shows up lighter gray. So it's hard to see, but the lighter gray there is kind of heart-shaped or kind of like bunny ears. That is the iohexol. So we knew that not only were these xylem conduits full of water, they were actually moving water with the heavy iohexol up the plant. So it was not just full of water, but actually functional. Well, this whole time series showed us two things. First of all, desiccation in the stem is a reversible process. And second, specifically, the xylem tissue, the water conducting tissue, was the first to dry out and the last to rehydrate. Well, this was pretty interesting in and of itself. But again, I always want to relate the structure more closely to the function. So I asked specifically, are there stem anatomical traits that facilitate reversible desiccation? And part of this came from an observation that some of these resurrection ferns have chloroplasts near the stipe xylem in that stele. Because to us, it seemed kind of surprising, like, why are there so many chloroplasts in the middle of the stem? Like if you have stem chloroplasts, you might kind of think, well, shouldn't they be on the outside where it gets to be more sun? So we wondered if that's important. And we also just searched generally using light microscopy, fluorescence, and staining for other important traits. Here's an example of the use of autofluorescence. Autofluorescence is a phenomenon where you shine certain wavelengths on the cool chemicals in plant tissues, and they shine back light from a longer wavelength at a slightly different color. So what's nifty about this image is there's actually no staining in this image. We used 3 channels, so 3 different images where we shined a certain wavelength on the plant cross-section, and it shined a certain different wavelength back. And we stacked those 3 images to make one colorful image. And the fluorescence signal we receive can tell us a bit about the chemical composition of the different tissues, which can be really informative. So for example, looking at a close-up image of the steel, the xylem conduits, which typically are moving water, were autofluorescing aqua, which is a combination of blue lignin, lignin's tough, and yellow, which could be cellulose or pectin. Which is a nifty finding because it means that these conduits are both tough, but not as tough as flowering plant xylem conduits because they're flexible. Pectin is flexible, so that might be helpful with changing shape during the desiccation process. This red tissue in the phloem and parenchyma would be chlorophyll fluorescence. Again, wondering why are those chloroplasts in the middle of the stem, and likely. This structure here is probably yellow from the suberin, which is a waxy layer because these ferns have a Casparian strip surrounding the vascular bundle, which might help keep water inside the vascular bundle during the resurrection process. We can also compare, this was a picture taken when the fern was desiccated, and then when the fern was hydrated. And, right off the bat, you can see some changes, right? The whole stele expands, just like we saw in the micro CT. Those xylem conduits change shape a bit as they expand. The chloroplast in the phloem has increased fluorescence, and the Casparian strip, that waxy layer, moved and expanded with the vascular bundle. So that's pretty nifty. And let's take a look at some of the other specific tissues. First of all, let's take a look at that cortex. Remember how the cortex dried out in development, and we were confused because normally the cortex stores water. Well, we found some cellulose and some lignin, lignin again being blue. And so it's pretty interesting to find a really strong lignified cortex. But then we realized that actually might make a lot of sense, because if the cortex dries out in development and it's not got water's turgor pressure to keep it strong, you need some tough lignin in the cortex. So maybe that lignified cortex is helping the plant stay strong and upright even when there's no water. We also did stain for lignin with fluoroglucinol HCl. This is bright field and this is fluorescence. And you can see the red lignin showing up here in the xylem conduits and in the parenchyma around it. Again, confirming the presence of lignin. And then we also stained for glycoproteins. And we used acid fuchsin, which shows up pink. And we really need more research to follow up and understand this better. But it was kind of exciting because it appears that the xylem conduits are lined with these glycoproteins, which, by the way, this particular stain fluoresced quite well too, which we think it might confer some flexibility or possibly helping these conduits be attracted to water, which might help the resurrection process. But we need more research to follow up and understand that better. Well, overall, let's just kind of tie together the structure and the function. Because ultimately, I was asking, like, how does the chemical composition that we can infer from the fluorescence relate to the function we saw in the micro CT? So, for example, the micro CT showed us that the cortex surrounding the vascular bundle dries out during development. And the histology shows us that the cortex contains some lignin, which helps explain how it can be so tough and supportive, even though it's got no water in it. The histology also shows us a Casparian strip which is waxy and prevents the flow of water, separates the steel in the cortex, which could explain why the water stays in here during resurrection instead of leaking out to rehydrate the cortex too. That might be adaptive because it might help the water move upward the plant to the leaf instead of leaking out to the cortex. Next, let's consider the phloem and parenchyma tissue. The micro-CT shows us that the phloem and parenchyma resurrect before the xylem refills, and the histology shows us that the parenchyma contains chloroplasts, which might have, they have a lot of sugars in it. It might osmotically attract water early in the resurrection process. And finally, let's think about the xylem. The xylem is compressed. The micro-CT shows that the xylem got compressed when desiccated and then had to change shape a little bit when it resurrected. And histology showed us that the xylem cell walls might be more flexible due to reduced lignin and the presence of flexible pectins and glycoproteins. So all in all, which just shows us that micro-CT, supported by the histology, is showing us that the site desiccation is a reversible process likely facilitated by some unique anatomical traits. And with that, I'd like to thank my funding sources, especially Pepperdine University, UC Santa Cruz National Science Foundation, and the Southern California Research Learning Center. A huge thanks to Steve Davis for a lot of the photographs and Dr. Anna Jacobson for a lot of the micrographs. And I received some incredible mentorship from the folks listed, and especially my PhD advisor, Dr. Yarmila Pitterman. And I would just want to say a huge thanks to all the students who helped me along the way. This is a photograph of my lab this summer and the students who worked with me. With that, I will take any questions. Thanks so much for listening.
On. Great. Oh, sorry about that. It took me a little while to find all the buttons. Yeah, thank you. That was like really informative about all kinds of stuff. And I love the fact that it was such a sort of varied, diverse set of approaches. Let's see. If anybody has questions, if anyone has questions, you can just unmute yourself and ask, or a couple of people are putting things in the chat. Josh Durr says, such a nice body of work. Thanks, Helen. Andy Simpson says, it has been suggested that prior to the rise of angiosperms in the Cretaceous, savanna/desert habitats were dominated by ferns. Can you weigh in on how plausible you think this is? If this hypothesis is correct, then I would predict that the drought-adapted ferns would be outside of the Polypodiaceae, since they diversified in the Cretaceous alongside angiosperms. What taxonomic groups do the ferns of the Santa Monica Mountains belong to? That was kind of a lot of questions in there.
Oh, this is great. Thanks, Andy. That's so exciting.
But there's a long follow-up after that, so I'll let
the long follow-up, but it's a different question.
Yeah, okay.
I'll take a look at that too. Yeah, so those resurrection ferns in the Santa Monica Mountains are mostly in the Chilanthoideae, which will be in the Pteridaceae. As I recall, remembering the Schneider et al. 2004 paper where they talk about ferns diversifying in the shadow of angiosperms, I believe the Pteridaceae were also one of the groups of ferns that diversified in the Cretaceous. So I guess the way I would interpret that is that they would also be one of the groups that radiated following angiosperms, but let's see.
That probably throws my hypothesis out the window.
Well, but I wouldn't say, honestly, I guess the short answer is I don't really know. I have to think about it. But what do you think?
I don't know. I'm asking you.
Yeah, let's see. That would be, I'd be curious to see the papers on that. That'd be, if you'd be so kind as to send me the citations, I'd be so interested to read more. I hadn't heard about those being dominated by ferns.
Wow. It's an idea that's been around for a while. I'm not sure I could find you papers for it.
No worries. but yeah, that's I want to learn more about that. That's right up right in my interest.
Do you want to go ahead and ask your second question rather than having me read the whole thing out?
Sure. Isoetes is a cam plant. It turns out that a fair number of aquatic plants are also cam. However, it's been suggested that Isoetes was a cam plant, or is a cam plant because its ancestors were cam plants, and its ancestors are trees with very little xylem as adults. And it occurs to me that your experiments on root pressure might signify that the roots might have been able to push water up into the tops of these trees despite the limited cross-sectional area to force water up. What do you think that this
that's a great question. Yeah, but I'll tell you what, honestly, I think root pressure probably wouldn't have helped very much in those tall lycopods. So that's one of the main hypotheses for why resurrection plants are so small, is that root pressure typically doesn't get you very high at all. So yeah, so I would suspect that those lycopods probably weren't using root pressure. That's a great question though.
Yeah, so there's a lot of love for you in the chat that I'll let you read, but there are a couple more questions. Joe Rigney says, is it possible that the leaf water provides the resurrection of the upper portion of the plant rather than water pressure?
Ooh, that's a great question. Well, here's honestly what I suspect. So in the targeted irrigation experiments, the root water when it was root water alone, it did get to the leaves eventually. But when it was leaf water alone, the leaves resurrected faster. But that makes sense, right? Because you're applying it directly on the leaf instead of waiting for it to come through the roots. And then when we did the experiment where we wet leaf, dry leaf treatment, where all the roots were getting wet, the wet leaf treatment resurrected faster. So I would suspect in that case, the immediate resurrection response is coming from the leaf water that's applied directly to the leaves. And then the root water ultimately is restoring hydraulic flow through the vascular system. So that's what I would suspect, is that the fast response is the leaf water, and then the root water is restoring the hydraulic flow. But I think, Josh, you have your hand raised?
Yeah, I was going to ask a question about embolisms. And do you think embolisms are forming in these stipes as they're desiccating? And how is that being repaired in the resurrection process?
Yes. Oh, so interesting. Well, actually, I'm this one I'm fairly confident the embolisms are forming, because in the micro CT, those air bubbles that were in the those black circles in the xylem conduits, those would have been air bubbles. Yeah. So I think yeah, so in that case, since that one I felt pretty good about, because that was an intact plant, so we hadn't cut it open and potentially introduced air embolisms, but we saw them there. So that I feel pretty good about, that there were likely air embolisms there. And then I think that the root pressure is likely the main mechanism, because we did find that in the coffee fern, you could resurrect some of the lower leaves with just capillary action alone, because when you just stuck the frond in a tube of water, capillary action was enough to get water and repair some of those embolisms and then get to the leaves, the lower leaves, but it wasn't enough to get all the way to the top. It's like the top leaves only resurrected when you push water into the frond. Yeah, so the short answer is yes, I think they're air embolisms, and I think that it's a combination of capillary action and root pressure that's repairing them. Does that make sense?
Great.
That's great.
Moby, did you have a question?
I did. I was wondering if you know, and if you do, great, and how, and if you don't know, is there a way to know when you're delivering the water to the root zone, for example, or even to the leaves, right? How do you know that you've delivered the sufficient amount to fully rehydrate the plant? So how can you determine, how do you know for sure that it's root pressure and maybe the pressure can't push it further, or maybe the water ran out?
Ooh, no, that's a great question. So I did a subset of experiments where I actually measured the water potential of the soil in different tissues that I was irrigating. So it made me feel so good about life and physics because it all made sense. So like the water potential of the soil was like minus 0.1 megapascals. And then the water potential of the rhizome was like minus 1 megapascal. And then the steel was like minus 1.5 and the leaf was like minus 2. So I was like, like physics works. So that's why I felt pretty good. And by the way, what was fascinating is that then I peeled off the cortex that was surrounding the steel, and it was like minus 50 to 100 megapascals. So it was kind of confirming that the cortex was staying dry while the steel in the middle was hydrated. And I was like, okay, physics works. So that was, but I will say it's hard to be entirely confident for every single one of those experiments, but the subset of experiments is kind of the best that I would do. And I felt better about it too, because since I knew that the rhizomes are shallow, the rhizomes are like 2 centimeters deep, and all the roots were pretty much in the top 10 centimeters. So yeah, so that's where I took my soil measurements.
Another question, really more about the CAM. Could it have anything to do with oxygen being aquatic, right? Is the oxygen more limited, and maybe crassulacean acid metabolism is helping it with the oxygen somehow?
You mean for isoides?
Yeah.
Ooh, well, it's because these ferns that I worked on weren't aquatic. But it would be interesting to think about isoides. Yeah, the short answer I think is that I don't know. What I desperately want to do is like invent some tiny little CO2 sensor to drop down in the xylem. And I just haven't, I don't know of that kind of technology at the time being. But yeah, the short answer is I don't know, but it'd be so interesting to think about, especially if the chloroplast near the xylem, especially thinking in the steel where you had that spike in glucose and fructose on the second day of resurrection, are they providing a little extra ATP that's helping with metabolic recovery in some way? I would just give anything to know exactly what's going on in there. But that's a great question.
On that, I am dropping a link to an article about the survey of CAM in submerged aquatic macrophytes in New Zealand.
Nice.
Most of which I don't think are isoides. I think there are a lot of angiosperms that are aquatic and are CAM plants, and maybe that paper and following leads in that will shed light on that question.
Thanks so much for sharing. I can't wait to read it.
Because I know I'm not an expert in any of this stuff by any means.
It's so interesting.
But I know of CAM more in the desert plants, right? And so,
yeah.
But thinking of the aquatic environment is potentially an oxygen issue.
Yeah.
Suffocation, lack of oxygen, somehow. I didn't know if there was that was a different way for it to help.
Yeah. Well, that'd be so interesting. And and I haven't worked on isoides at all yet, but that'd be so interesting. And I have to say, I haven't measured gas exchange in these ferns at night. So that could be an interesting. Okay, though I did one pilot experiment where I resurrected them in the dark to see if that made a difference. And it didn't really make a difference. Got pretty much the same resurrection. Because I was like, ooh, maybe that's it. But I'd love to follow up at some point.
Yeah, so Mickey Long put a question in the chat that I think is a really interesting one. Do you want to throw it out there for us?
Sure.
Well, I'll why don't do you want to, Mickey Long, do you want to go ahead and make it public?
I'm fresh off of reading Suzanne Simard's book on the mother tree and the incredible mycorrhizal functions. So what do we know about fern mycorrhizae and their help sending water nutrients to the plants?
That's such a great question. Oh my goodness. I think the short answer is I don't know. I don't think we know that much. And so I'm starting to collaborate we have a microbiome scientist here at Pepperdine, and we're starting to collaborate on the fern microbiome, because honestly that was one of my first hypotheses for how the resurrection ferns might resurrect so well. I thought, because like they have those shallow roots, maybe they're getting help from mycorrhizae. So kind of a pet experiment that I've been dying to do is to resurrect the ferns with a fungicide and see if they can still resurrect if you know that the mycorrhizae aren't functional. But I just haven't got around to it yet. But I sure wonder, especially because it was a little embarrassing when I started these experiments. I had a lot of failed resurrections, and I found that if you transplanted the ferns while they were hydrated and active and then dried them out, they would resurrect. But if you went out there and dug up a fern that was already dried up and tried to resurrect it, it wouldn't resurrect. And my main hypothesis for that is that somehow the root system, maybe also the mycorrhizae, get disturbed if you transplant it from it while it's in a desiccated state. So the short answer is I don't know, and I'm just dying to know. I think it'd be so interesting.
I can't shed any botanical info on this, but we did collect a lot of xeric ferns actually in Oaxaca in Mexico.
Oh, nice.
In the late 80s. And we were there in January during their dry phase. So everything was shriveled up. I learned everything from looking at it curled up in their hairs and scales. And then when they all resurrected back in New York months later, I had to relearn them all because I couldn't recognize anything because they looked completely different. But anyway, we collected them. All of those were really collected in their dry state. We collected them carefully, transported back to New York, but those were potted, and really most of them survived. So I can't say more than that, but I don't know.
Yeah.
Say maybe don't give up. I don't
know. And I should add the caveat. I didn't quite explain that right. I should add the caveat. They always resurrected when I dumped the leaves in water. But what seemed to be broken is if I just watered the roots, then the water wouldn't go from the roots to the leaves to resurrect the leaves. But you're right. When I transplanted potted plants that were already dried up, if I dumped water on the leaves, always they'd resurrect. But yeah, but that's so interesting. I've been dying to go to Oaxaca. I had a friend in Oaxaca who told me I just have to go there to see the resurrection ferns. So.
Don, let me just jump in and say, excuse me, we're actually, kind of out of time. Anybody who, has other obligations, feel free to jump off. But there are still more questions coming. So anyone else who wants to stay and continue the conversation, like Michael Simpson, apparently.
Okay. Hey, very good talk, Helen. That was great. I was struck by the detachment of the endodermis and the kind of collapsing of those cells. I guess I would have assumed that the whole mechanism would have been non-functional, after that. And you kind of implied maybe it's a chemistry thing. So do you have any comparative data with these resurrection ferns, with something like Selaginella, with other vascular plants? Do other plants do this? Or is it just a resurrection thing? Yeah.
Oh my goodness. I am just dying to know the answer to that too. I just had a student project in my botany class this spring where they tried to do exactly that. The caveat is they had a really small sample size. So I had them compare, because they only had 3 weeks for their project, right? So they compared a resurrection fern with a non-resurrection fern in the Pteridaceae, and then in the Polypodiaceae, a resurrection, non-resurrection fern. The short answer that the very, very little preliminary data we got that surprised us is that it was similar in the resurrection and non-resurrection ferns. And I was like, what? I was blew my hypothesis out of the water. So yeah, the short answer is I don't know, but
So
I want to do more. Yeah.
Is it a fern thing, or is it,
yeah.
Has this been investigated in other plants? No?
Not to my knowledge, no. Okay. Yeah, so I'm curious. I'm definitely planning to follow up because, I only looked at 4 species. Yeah, it might be that the overall, all right, because it's kind of weird where it seemed, at least in the one, it was Adiantum jordani that we were comparing, and it seemed like the whole stele detached kind of from the cortex there as well, but not as cleanly as it did in Pentagramma. Pentagram is actually kind of fun. Like if the stipe is dried up and you have a little piece of it, you can actually just pull the whole steel out. And it doesn't happen quite as cleanly in Adiantum jordani. So maybe, maybe there's some like smaller level chemical difference that makes it different.
Good. Yeah, I hope you do comparative studies with I don't know if this has been done before.
Not to my knowledge. I'd be curious to hear about it if anyone knows anything. But yeah, I'm just dying to know. The tricky thing, of course, is finding closely related desiccation tolerant and non-desiccation tolerant ferns, which actually that's a whole, like my original idea for my PhD was to, drive around and water all the ferns and see which ones are resurrection ferns, because it's actually kind of hard to have a definitive list of what's a resurrection fern and not, because you don't publish negative results, right? If a dead plant stays dead, no one writes a paper about it. So it's anyway, but thanks so much. Yeah, I desperately want to know the answer to that too.
Okay, very good.
We have a couple in the chat here. Yeah, from Patty Litton and Peter Ralston. What about chloroplasts embedded in the steel? What function would they perform?
That's such a good question. Yeah, it's such a good question. I don't know. And one hypothesis is that since those chloroplasts would have a lot of sugars, maybe it, especially because we saw that the phloem resurrected first in the micro CT, maybe all those sugars like osmotically attract water into that tissue and help pull water in through osmosis. The other thing that's a little bit more nebulous that I really don't have much data to support, but I just wonder if there's some more metabolically active pumping of water that's facilitated by energy from the chloroplast. But that's pretty speculative of me to even say that at this point. That's what I still not quite sure how to measure.
So is that a common phenomena that chloroplasts appear in various places in plants, even when no photosynthesis could be occurring in that location?
To my knowledge, not so much, because I was starting to read about, and there's some interesting, actually in salt tolerant mangroves, there's some interesting studies on xylary chloroplasts. But to my knowledge, there's not a lot of work done on that. I'd be so curious to do more experiments and learn how it worked.
That'd be cool.
Is it at all possible that sunlight could be penetrating?
Oh my goodness, that's the other thing I really want to know, because I was trying to picture how I could do this, because it's just so tiny, like the whole stipe is 1 or 2 millimeters in diameter. I was thinking, what PAR sensor is like a quarter of a quarter is that narrow that I could find out? Because honestly, I have no idea if light's getting through.
We have another question here. When fire burns everything above ground, what happens?
Oh, that's so fun. So my student was working on that this summer, actually, because, in 2018, we had the Woolsey fire burn half the Santa Monica Mountains. And it's so fascinating because we think there's differences in how the fern species respond to fire. Most of the fern species do just fine, I think, because the rhizomes underground. So you see resprouting essentially, where the rhizomes survive the fire and it just sends up new fronds. Those fronds might have differences because now they're in a full sun environment for a couple of years while the chaparral comes back instead of a pretty shady environment. So, you have frond angles pretty steep. The leaf morphology is different. It's sun leaves instead of shade leaves. But what was fascinating is at one site, so I'm not sure we have the replication to demonstrate it, but at one site, most of the pentagramma, those resurrection ferns, were wiped out. And we think it's because they have those really shallow rhizomes. I know I counted at one point, there were over 100 at this one site. And then after the fire, there were 2. And they haven't really seemed to recruit back yet. Having said that, though, one of the other post-burn sites that's more rocky and there was less fuel load, a lot of pentagramma did survive the fire. So I think it's affected by, how hot the fire was, how much fuel there was, and so how deep the heat penetrated into the soil. But I think the resurrection ferns would be most vulnerable because they have such a shallow rhizome.
Could it be, doesn't the pentagram is the goldback? I mean, is waxy?
Oh, that's a good question.
Could
You know,
that be more flammable and make it burn faster, hotter, stronger?
yeah,
Something?
you know, that's a good question. That'd be a fun experiment to do in the lab. I kind of suspect, I'll be honest, I kind of suspect that the chaparral burns so hot, it might, you know, it might not matter, but that'd be really fun to find out. I didn't see, you know, after the fire, it was just like a moonscape. It was like, you know, not a single piece of above ground vegetation appeared to be there.
Well, if you're going to be torching ferns in the lab, let me know. I always like a good torching event.
Yeah.
Andy Simpson, a question?
Yes, question. On the subject of shade leaves versus sun leaves in ferns following fire, Kevin Boyce did a study on shade leaves and sun leaves in large angiosperm trees and found, or seemed to find, that the distinction between shade leaves and sun leaves is not the amount of sunlight, but rather the amount of water that is making it up the stem. Which I would not expect to be the case in not expected to matter in ferns. Did you see a difference between shade leaves and sun leaves anyway?
You know, that's such a great question. The short answer is we haven't really tested it carefully yet. Yeah, because she did such a broad ecological study. We went to all these sites and all she measured really was frond angle. And we didn't do any anatomy, but that's such a great idea. We should follow up and compare the anatomy. Yeah, I'd be so curious. Well, and we should follow up and compare the water availability. Well, we did like temperature and relative humidity, but it'd be interesting to see soil water availability. But yeah, thanks for the idea. That's a good one.
Okay, any other questions? I'll save the chat and send the whole thing to you so you can see the other comments that people have made. Basically, everyone thinks it was a great talk.
Well, thank you guys all so much for the invitation, and thank you for the great questions. I had so much fun. You guys are a fantastic group. I learned so much from you guys, so thank you so much.
There is a one final question here. So how deep are the fern rhizomes?
Oh,
Because
yes.
apparently, Hal says with their dicentra uniflora and passiflora, 2.5 centimeters deep is critical for fire survival. Wow,
Oh, that's so consistent with so consistent with what we found. Because the pentagramma and we measured the rhizome depth of all of them, and I think that was in my 2016 paper. But pentagramma was like 2 centimeters rhizome depth with 8 centimeters root depth, and then Palea I think was a little deeper than 2 centimeters for the rhizome depth, and then 12 centimeter root depth. And that would be consistent with like at that one site we had a lot more Palea survive the fire than pentagramma. So maybe it's just being under that 2.5 centimeters might be why so many of them got burned up. So that would be actually, Hal, if you don't mind sending me that that citation, I'd love to read that because that would be really consistent with what we're finding. But yeah, thanks for the great question.
This has been super informative all around. Final questions for those of you. I mean, this is, you know, not only was it a topic that I had actually wondered myself, but also, you know, you're using a lot of techniques that I personally know like nothing about. So it was really, for me, really a nice introduction to some of these things. So
thank you. Yeah, this was so much fun.
Okay. Looking, I don't see any more questions here. Yeah. Any other questions from the people left on the Zoom? Going once, going twice.
Fabulous. Thank you.
Okay. Yeah.
Thank you so much. And anyone who wants to follow up and chat more, I'll put my email in the chat. Don't hesitate. Please feel welcome to send me an email. I'd love to talk about anything more. I just appreciate y'all's time so much. This was such a fun conversation.
So, if you can stay on for just a second, Helen, I just want to discuss one logistical thing when everybody else, or at least, I mean, if people want to hear the logistical thing, they're welcome to, but, so, yeah, basically what happened was we had a little connectivity problem.
Oh, no worries at all. No worries.
No, so I think the issue is that there's probably there might be gaps in the recording, and so we might want to ask you to like re-record some little bits to try
Oh,
to
sure.
yeah. So
Yeah, absolutely.
it'll take a little while to sort it out because, you know, as I mentioned, I'm still in effing Anchorage and a little bit kind of limited in my ability to do things. But when I get home, you know, I can see how much I have. And Mitchell was recording the first part, and I can take what he has, and we can try to put it together and see what's missing.
Yeah, sounds great.
All
I'd
right.
be happy to re-record anything.
Great. Thank you so much. Thank you so much. That really was just a fascinating talk. And because there were a lot of logistical problems, there were times when I wasn't able to pay as much attention as I wanted. So I'm actually looking forward to the recording so I can see and hear the whole thing. And I know Josh missed the beginning, and I'm sure he wants to hear that also.
I've seen a version of this talk.
Yeah, I was gonna
before.
say, I felt bad. Josh, when I saw you, I felt bad. I was like, there's definitely seen a little of this before.
No, this was great, Helen. I'll be in touch. I want to come out and visit you, and let's talk about a proposal.
Yes, please. That'd be so fun.
Great. Awesome. Anyone else have any final questions or comments? Moby, you guys should exchange information so you can tell her how to grow the ferns.
Well,
So,
well,
That'd be great.
yeah, all right. Well, I'm gonna end the Zoom unless people want to keep chatting.
Thank
Thanks
you all so much.
Thank you.
so
See
much.
you later.
Bye. Great to see you, Moby. Great to see you.
Great to see you, Amy. Thank you.
Bye.